Hi, it's Dr. Centeno, and today I'd like to talk to you about what is cell viability, how is it determined, and what does it mean? So if you wanted to understand one absolutely critical number that's being thrown around right now by amniotic and cord blood and other tissue vendors, it would certainly be viability. Technically, viability is a simple snapshot that just means the percentage of cells that are living. However, it doesn't really tell you the whole story because you know nothing about cell health or vitality. As an example, if we use patients here so that you can better understand it, all three of these patients are technically alive and would be counted as alive in a test of live dead. So looking at stem cell viability and vitality really looks more like this. There's this simple viability assay, which is quick and dirty. But then there's also an apoptosis assay, and also a cell proliferation assay, and also a cell function assay. And you have to have all of these to look at cell viability and vitality. So first, what tests are used to determine viability, and how do those work? Well, the most common type of assay is what's called a dye exclusion. You basically put in a dye with a bunch of cells, and the cells that are living will exclude the dye because they have an intact membrane, and the cells that are dead will take up the dye because their membrane is not intact. Now, that only really tells you about how the cell membrane is working. So common dye exclusion tests include trypan blue, eosin, and propidium. However, cells also have a process called apoptosis, which means they're on the road to dying. And apoptotic cells are often counted as viable in dye exclusion tests. Why? Because the plasma membrane is still intact in these apoptotic cells. And apoptosis is therefore measured a different way, usually by using flow cytometry to find a phosphatidylserine molecule that's put up on the cell surface. And cells use this as a I'm dying marker. So credible cell viability and health studies always include measuring the level of dying or apoptotic cells via flow cytometry. Another essential step to determine viability and vitality is to grow the cells in culture to see if they can form colonies and propagate. Obviously, dead and apoptotic cells won't survive and grow. And finally, for stem cells, you need a functional assay. And this is usually tri-lineage differentiation for mesenchymal stem cells to make sure that they can still function, i.e. become osteoblasts, adipocytes, and chondrocytes. So again, you need to expect that all four of these tests are run when looking at cell viability and vitality. Not just the first one, which is what you usually see, simple viability. So for more information, see regenexx.com and have a great day.